Let us now go to the planes of the body. Sagittal divides the body into right and left parts. Transverse or horizontal divides the body into superior and inferior portions. Frontal or coronal divides the body into anterior or posterior parts. There are body cavities such as mediastinum, diaphragm, and pelvic cavity. Serous membrane is a membrane that lines the trunk cavities. Mesenteries are specialized membrane in the abdominopelvic cavity. Let us now move on to the cytology. It is the study of the cells. Organelles are subcellular structures specialized for specific functions. The nucleus is known as the control center or headquarter of the cell. Ribosomes are the site for protein synthesis. Rough ER has ribosomes while smooth ER has no ribosomes. Golgi apparatus modify, package, and distribute lipids and proteins. Mitochondria is the powerhouse of the cell. Lysosome is the digestive system of the cell. Peroxisome eliminates toxic substances, and cytoskeleton gives framework. Cilia propel substances along the surface. Flagella move an entire cell, and secretory vesicles deliver the proteins to the plasma membrane. The plasma membrane acts as a barrier and regulates what enters and leaves the cell. According to the fluid mosaic model, the membrane is a mosaic of proteins floating like icebergs in a lipid bilayer sea. Lipid bilayer consists of two back-to-back layers of phospholipids, cholesterol, and glycolipids. There are two types of transport systems across the cell membrane. Passive transport is also known as downhill movement or a movement from a high concentration to a lower concentration. Diffusion is a random mixing. Optical diffusion is the kinetic movement of molecules and does not use carrier proteins. Facilitated diffusion requires ATP and facilitated by carrier proteins. Second is active transport or uphill movement from a lower concentration to a higher concentration. The two types are primary active transport and secondary active transport. Let us now move on to the protein synthesis. It is the formation of proteins using gene expression.